Today we are going to study about Clonorchis sinensis. So the parasite Clonorchis sinensis is also commonly known as Chinese liver fluke or Oriental liver fluke. Now talking about the zoological classification of Clonorchis sinensis, it falls under the kingdom Animalia, phylum Platyhelminthes, which includes the worms that are flat in their structure, morphological structures, and class Tremetroda, which includes different kinds of flukes, order Opisthorchidae, family Opisthorchidae, genus Clonorchis, and species Clonorchis sinensis. Here on the right side, you can see the morphological form of the parasite Clonorchis sinensis. These are the two morphological forms. One is the adult worm, and another is the egg. So, are you getting me?
Yes, ma'am.
History and geographical distribution of Clonorchis sinensis. The Clonorchis sinensis was first described in 1875 by McConnell in the autopsy material of a Chinese carpenter in Calcutta Medical College Hospital. So since the worm was first encountered from the autopsy material of a Chinese carpenter in the biliary tract, so the worm was also commonly named as Chinese liver fluke. And the life cycle of Clonorchis sinensis was first described by Faust and Kaw in 1927. So the parasite Clonorchis sinensis is mostly endemic in those countries where there is poor sanitation. So this parasite is common among the population who eat raw, pickled, smoked, dried, or salted freshwater fish. So this freshwater fish acts as the intermediate host for this parasite Clonorchis sinensis. Now the geographical distribution further, the parasite is mainly found in Asian countries. So since the parasite is found in Asian countries, that is the East World countries or the Oriental countries, the parasite was also named as Oriental liver fluke. So now the infection caused by Clonorchis sinensis is known as Clonorchiasis. The infection caused by Clonorchis sinensis is called as Clonorchiasis, and thus the Clonorchiasis mainly occurs in Asian countries. Among the Asian countries, it is mostly prevalent in the countries like China, Japan, Taiwan, Korea, and Vietnam. So the Clonorchiasis is endemic in these Asian countries, and in the China, it is mostly found in the southeastern part of China, where the fish are cultured in ponds and they are fertilized with human or animal fishes. So in the southeastern part of China, where the freshwater fish is cultured with the human or animal fishes, is the endemic area of this Clonorchiasis. So this human act as the definitive host for this Clonorchis sinensis. So the freshwater fish is one of the intermediate host, and the human are the definitive host for this Clonorchis sinensis. And in these Asian countries, it has been estimated that approximately 19 million people are infected with this parasite. So since this parasite is mostly common in the Asian countries, the Clonorchiasis infection, if occurs outside the Asian countries, these are mostly the imported cases. That means Clonorchis has also been found in United States, Canada, and Australia also. So in these countries, the it was found in the Asian refugees who have moved from these endemic areas. Uh, did you get me or not?
Yes, ma'am.
These imported cases means that the persons who have moved from the Asian countries to these other countries worked as a carrier for this clonorchiasis. So this is the diagram for geographical distribution of clonorchiasis. Here in the map, you can see that the clonorchiasis is mostly found in these Asian countries like Japan, Korea, China, Taiwan, and now talking about the morphological forms of the parasite. The Clonorchis sinensis has two different morphological forms. That is, one is the adult one, and other is the adult worm. Clonorchis sinensis has a flat, transparent, spatulate body which is pointed anteriorly and rounded posteriorly. And the worm has a size of 10 to 20 millimeter long and a breadth. It is 10 to 20 millimeter long and 3 to 4 millimeter broad in its morphological form. And it has two different kinds of suckers. One is the oral sucker and another is the ventral sucker. The oral sucker is slightly larger than the ventral sucker, and the ventral sucker is located at the junction of the anterior and the middle third of the body. And it consists of intestinal cica, which is blind, simple, and extend to the caudal region. The parasite is a hermaphrodite worm, and it has two testes which are large, extensively branched, and lie one behind the other in the posterior third of the body. Here in the diagram, you can see there are two testes. One lies above, and another lies behind the testis. And the hermaphrodite worm discharges eggs into the bile duct. And the mature Clonorchis sinensis worm discharges 2,400 eggs per day as found in cats. In cat, 2,400 eggs per day per fluke has been reported. And in one instance, 21,000 adult flukes were reported from the autopsy material of a man. So in cat, 2,400 eggs per day is discharged by the adult fluke, and in the man, that is a definitive host, 21,000 adult flukes were reported in one case. And the worm has a lifespan of 25 to 30 years. So morphological now, the another morphological form is the egg. The egg is broadly ovoid or flask-shaped, and it has a size of 35 micrometer by 20 micrometer. It is yellowish brown. It is bile stained. Thus, the color of the egg is yellowish brown. Its cell is bile stained, so it has a yellowish brown color. And the egg is operculated at the anterior side, and in the posterior side it has a hook-like spine. So in the diagram you can see in the lower left side there you can see that the labeled part there, the egg has a operculum at the anterior and a tiny knob or spine at the posterior side. And the egg, due to the presence of operculum, it doesn't float in saturated salt solution. So due to the presence of operculum, it provides the egg buoyancy, so it doesn't. Due to the presence of operculum, it does not float in saturated salt solution. So when the egg is laid, it bears a ciliated embryo, that is miracidium. So in the when a definitive host discharges the eggs along with the feces, the egg consists of ciliated embryo, that is miracidium. And the egg, though it is embryonated, it does not hatch in water. So it hatch only when it encounters the intermediate mollusk host. So the egg hatches inside the body of the mollusk host, intermediate host. So the egg is infective only for the mollusk. So as it hatches inside the body of mollusk host, it is infective to the mollusks only. Now, life cycle. The Clonorchis sinensis completes its life cycle in 3 different hosts. And among the 3 different hosts, one is the definitive host, and it completes its life cycle in 2 intermediate hosts. And among the definitive hosts, like the man, dog, the definitive hosts are man, dog, or cat. And the man is the principal host, and on other freshwater fish-eating canis like the dogs or cats act as the reservoir host. So man is the principal host, and dog and cat act as the reservoir host. And the first intermediate host for this Clonorchis sinensis is the snail, especially of the genus Bulimus, Paraphosarulus, and Alosinma. So the first intermediate host is the snail. And the second intermediate host for this worm is freshwater fish of the Cyprinidae family, which includes the crab fishes. So the life cycle completes in 1 definitive and 2 different intermediate hosts. And the infective form for the parasite is Metacercaria larva, which is found in the flesh of freshwater fish. So the life cycle of Clonorchis sinensis is perpetuated by humans or reservoir hosts defecating in or near freshwater sources containing suitable species of snails. So this life cycle is maintained by the human or reservoir hosts who defecate near the freshwater sources containing suitable species of snails. So once the eggs are defecated with the feces in the fresh water, the eggs of Clonorchis sinensis containing miracidium are passed. Eggs of Clonorchis sinensis containing miracidium are passed with the feces of definitive or reservoir host in the fresh water. Now, in the fresh water, though the egg is embryonated, it do not hatch in water. So the egg hatches only when it encounters a suitable intermediate host, that is the snail. So once the eggs are ingested by suitable species of snails, the eggs hatch in the midgut of the snail and releases the miracidium larva. So now the miracidium penetrate the intestinal wall to reach the vascular space. Now it reaches the vascular space. Now in the vascular space, the miracidium develops through the different larval stages like the sporocyst, and then it develops into radia stages, and lastly it becomes Lophocercus Cercaria with a large fluted tail. So it through the miracidium it develops into sporocyst, then into radia, and lastly into Lophocercus cercaria. That is, it has a single tail. So it develops into cercaria, and this time for development into cercaria takes up to 21 days, that is 3 weeks. Now the cercaria escape from the infected snail into the fresh water, and it moves freely in the water, that is it swims in the water, and as soon as it encounters a suitable second intermediate host, that is the crayfish, it penetrates the skin of that fish. So here the egg bearing miracidium was now converted to was developed into cercaria, and now cercaria has penetrated into the skin of its second intermediate host, that is the crayfish. Now, as it penetrates the skin of the crayfish, the cercaria sets its unforked tail. So it sets out its tail, get incrusted under the scales or the flesh of the fish to become metacercaria in about 3 weeks or 23 days. Now, this metacercaria is the infective stage for humans. So, when a human eats raw or undercooked freshwater fish bearing this metacercaria cyst, then the human becomes infected with this parasite. So, the metacercarial cyst measures 140 micrometer by 120 micrometer in size, and a single fish may contain as many as 3,000 cysts. So now the mode of infection for man is the man acquires infection by eating raw or undercooked freshwater fish carrying metacercaria larva. So this is the infective form for human or other reservoir host. Now, the infection may also occur when or cooking utensils during preparation of the fish for cooking. So, one mode of infection is by the consumption of raw or undercooked crayfish, and another is through the contaminated fingers or cooking utensils during preparation of the fish for cooking. Now, once the metacercaria has been ingested by its definitive or the reservoir host, the metacercaria exist in the duodenum, and the liberated young flukes or adult that come out, these enter into the common bile duct through the ampulla of Batter. So they exist in the duodenum, and through the ampulla of Batter, these adult cercaria get entry into the common bile duct. And in the after entry into the common bile duct, they proceed to the distal bile capillaries and get mature. And this worm becomes mature in the distal bile capillaries in about a month. That is 25 to 30 days. Adult worms, these when mature, they produce on average 10,000 eggs per day. So once the eggs are liberated, the eggs exit the bile duct and are excreted with the feces of definitive and reservoir host near or in the freshwater, and then the life cycle is repeated. So this is the life cycle of Clonorchis sinensis. Here the in the diagram you can see the embryonated eggs passed in phases number 1, then it get it is ingested by the first intermediate host, that is the snail, and in the snail it develops into the Cercaria larva, and the Cercaria then gets insisted into the scale or flesh of the freshwater fishes, and develops into metacercaria larva. And when the human, that is a definitive host or the reservoir host, consumes the undercooked or raw fishes, they harbor the metacercaria larva and or the worm, and it develops into adult in the definitive host. And the adult worm begins to produce egg, and the eggs are passed to the fishes, and the life cycle is repeated again. Now the pathogenesis of Clonorchis sinensis. Here in the life cycle, we understood that the Clonorchis sinensis inhabits in the distal bile capillaries. And in the distal bile capillaries, adult worm, the metacercaria larva develops into the adult worm. And here in the distal bile capillaries, the adult worm feeds on the ductal epithelium, and it produces different kind of the worm produces different kind of toxins, which results in mechanical irritation of the definitive host. And this mechanical irritations results in the inflammatory response of the duct epithelium. And due to the infiltration of different kind of inflammatory cells, there is the formation of pathogenic lesions. So the pathogenic lesions occurs due to the toxin liberated by the adult worm, which results in mechanical irritation and then inflammatory response. So now the pathogenic changes that occurs in the definitive host depend upon the intensity and duration of infection. So the pathogenic lesions may be either light or heavy type. In the light infection, there is only mild inflammatory response of the ductal epithelium. So, and there is either no change or only little change in the liver parenchyma. So in light infection, that is when the adult worm are present less than 10 in number, then there is only mild type of inflammatory response and little or no change in the liver parenchyma. And in heavy infection, there is thickening and localized dilation of the bile duct. That is, there is cystic dilation in case of heavy infection, and there may be hyperplasia of the mucinous glands, and this can lead to neoplasm of the bile duct, and in some cases cholangiocarcinoma is also reported. So now clinical features. More than 70% of the light infections are asymptomatic. That is, there are no any symptoms in more than 70% cases of light infections. So in heavy worm burden, that is in heavy infection, that is when the worm burden is 100 to 1000, this leads to early symptoms like fever, diarrhea, epigastric pain, tender hepatomegaly, and anorexia. So in early stages, due to heavier worm burden, there may be fever, diarrhea, epigastric pain, tender hepatomegaly, and anorexia. So if the infection progresses, it results in chronic stages, and in chronic infection, it may result in occasionally biliary obstruction. Due to the heavy worm burden, there may be biliary obstruction, and in some cases jaundice is also reported, And the chronic infection may also result in calculus formation or stone formation. In some cases only, there is biliary cirrhosis and portal hypertension. And in some instances, cholangiocarcinoma is also reported. That is, the cancer of the bile duct is also reported. And in some patients with chronic clonorchiasis, it tend to become these patients tend to become biliary carriers of typhoid bacilli. So in some patients with this clonorchiasis, they become biliary carriers of the typhoid bacilli. Now the laboratory diagnosis of clonorchiasis. So the laboratory diagnosis of clonorchiasis is mainly demonstrated or diagnosed by the examination of feces, which bears characteristics eggs in the feces and aspirated bile. So for the laboratory diagnosis, it is based on the examination of characteristics eggs in the feces and aspirated bile. That is, the egg which is small, oval shaped, operculated, and has a operculum and a small tiny lobe in the posterior part is the characteristic egg of the Clonorchis sinensis. And while examination of the eggs or feces or aspirated bile, if you see such kinds of characteristic egg, then you can say that this is the egg of the Clonorchis sinensis. So for the demonstration of egg, two types of tests are usually done. One is the direct smear examination. Here, in the direct smear examination, saline preparation of the feces and or bile is done. And for this, 1 drop of saline is kept in a glass slide, and a loop full of feces is emulsified with the saline, and a cover sleeve is used to cover that drop. And it is observed on under 10 low power microscope, and then under 40x 100 power. Then you look for the characteristics egg. And this is usually possible in case of heavy infections. So in case of heavy infection, the eggs are passed with the feces and are also visible in the aspirated bile. So in the direct smear examination, we were talking about the laboratory diagnosis of Clonorchis sinensis. So for the laboratory diagnosis, we can their demonstration of egg is done, or there are other immunological tests available. So under the demonstration of egg, there are two different techniques that can be applied. That is the direct smear examination, and another is the concentration method. In the direct smear examination, either the saline preparation of feces or the aspirated bile is done, and this is possible only in case of heavy infection. In case of heavy infection, the saline preparation of feces and bile demonstrates the characteristics eggs. So in cases of light infection, the sedimentation concentration method can be applied. Here the feces is emulsified with common salt, and since the egg doesn't float in the saturated salt solution, the supernatant is discarded, and the residual part is taken as the specimen, and it is placed in the glass slide and observed on the microscope. So this concentration method is used for the light infection, in cases of light infection. So if there is biliary obstruction in the patient, the eggs do not appear in the stool. As the biliary duct is obstructed, the eggs remain in the gallbladder. So in these cases, the bile should be aspirated for the diagnosis of clonorchiasis. That is, the needle aspiration surgery or autopsy specimen is required for diagnosis in cases of biliary obstruction. So that was the demonstration of egg. Now the immunological tests are also available for the laboratory diagnosis, and under the immunological test, the immunological tests are not readily available. So, if available, the complement fixation test and indirect hemagglutination test are usually used for the laboratory diagnosis of clonorchiasis, in which the antigens of Opisthochus sinensis are used for the laboratory diagnosis, and this antigen react with the specimen and give positive result. And another immunological test is the intradermal test, which is available in some centers only. So intradermal test is the immediate type hypersensitivity test, which is available in some centers only. So it is not most frequently used for the laboratory diagnosis. So mostly for the laboratory diagnosis, the demonstration of eggs should be done by the microscopic method, or the by the direct microscopic method or the concentration method. Now, treatment. For the treatment of clonorchiasis, praziquantel 25 mg per kg 3 times daily for 1 day is usually prescribed. So for the treatment of clonorchiasis, praziquantel is the best drug of choice. So in case of obstructive jaundice or obstructive biliary tract, the surgery is done for the removal of the gallbladder. So this praziquantel causes paralysis of the muscle of the adult worm. So as a result, the adult worm um, cannot attach properly to the bile duct, and it is excreted out with the feces. So this praziquantel is used as the drug of choice for the treatment of clonorchiasis. So now talking about the prophylaxis of clonorchiasis. For the prophylaxis of clonorchiasis, the proper cooking of fish should be done, since the infection occurs when the fish is eaten raw or undercooked. For the prophylaxis, proper cooking of the fish should be done. And as the human and other reservoir host act as the definitive host for the sustenance of the parasite, proper disposal of human or other canis like the cat or dog feces should be done. So as this feces is properly disposed, it cannot encounter its intermediate host, and the life cycle is ceased. So proper disposal is a critical part for the prophylaxis of this disease. And the control of snail, which is the intermediate host. So if the snail is controlled, that is the population is controlled, then the parasite cannot complete its life cycle, and hence the life cycle is seized, and further infection is also restricted. So these are the prophylaxis for the clonorchiasis. Okay, thank you.